Welcome everybody to our PARC seminar, Pain and Addiction Research Center here at UCSF. So it's my pleasure to introduce Tom Kash today. So Tom is a distinguished professor and vice chair of Department of Pharmacology at UNC Chapel Hill. And he's had a very productive and influential career in a range of sort of interconnected areas, including serotonin and various aspects of nucleus striatum social behavior, all kinds of interesting things. And it's funny, he's giving a talk today which is entitled Examining the Extended Amygdala Signaling in Pain, Anxiety, Alcohol Drinking. And I always feel there's a sort of entertaining tug of war in neuroanatomy fields where people talk about the extended amygdala because they want to take over bits of the basal ganglia and things like that. Whereas the basal ganglia people like me are always thinking, yeah, central amygdala is always really just part of the striatum. So it's a little back and forth. Anyway, I'm really looking forward to your talk, Tom.
Wonderful. Well, thank you, Josh. It's great to get a chance to chat with some folks, see some familiar faces. It'd be great to be there, but also I'm just excited to share some of our work. All right, and yeah, so this is sort of a deep foray into some of the things the lab has worked on, and just sort of front end, I like to just want to acknowledge this is sort of been a long scoping project through the lab over the years. A lot of different contributors who are bolded here and call outs on many of the slides, as well as collaborators and our funders, primarily NIAAA, but also we have money from NINDS2 to work more on some of the pain work. And so here's our group, here's our center, and that's one of my goats, Irwin the goat, with his majestic horns. And so for me, I think, now more than ever, it's important to ground ourselves why we're doing this work. And, I'm at an alcohol center, and really, how my inroad to what we're studying really stems from alcohol and how that can impact health. And so, as many of you may know, alcohol abuse, alcoholism, alcohol use disorder, it is a tremendous public health burden, personal burden. Many people around the globe die. Estimates, 141,000 Americans die each year. There's a high prevalence of alcohol use disorder. If we think about alcohol-related deaths, there's, sort of a lot of health reasons, but also psychiatric reasons or accidents. So there's, it's not as if this happens in isolation. This is, each death you can think of, this is a person who has has friends and family, and this is ringing through their lives. And this is something to me that I think is important to understand, because I'd like to think this is a these are preventable deaths. And, it impacts health via alcohol use, really has negative impacts on many different systems. And so, if you think about just the whole body, there is a tremendous impact on nearly every tissue. We think, I'm a brain person, so I think a lot about brain and neurological issues, but this is far from the only thing. There's heart issues, bone issues, muscle, GI problems, of course, the liver. And so this is a real, alcohol is really bad for you. And I know this is maybe not a message people like to hear, but it is an important one. It has really robust bad effects on many parts of your body. But again, I focus on the brain. That's what, that's sort of where I come into play. And in part because the brain is important for driving our behavior. And when we think about alcohol in the brain, there's a lot of different comorbidities and interactions ranging from neurodegenerative disease or anxiety or pain or cognition. And, I think there's a lot of really exciting different aspects to understand, but I've really sort of focused my career, my question on the way I think of it is, are the bad things associated with alcohol use? We think of pain, anxiety, or stress, or PTSD, and I'm sort of I've always felt whenever I go to like an addiction meeting or something, there are all these folks who are really interested in reward, and I appreciate that, but I am really much more interested in why do bad things, how do they shape our behavior? And so really this has been where I've spent most of my professional life, sort of delving into these subtopics. And the idea that alcohol is really, there's critical bidirectional interactions with these various comorbidities. And just to highlight one, and this is from the animal model work, So we think about pain. And so this was a small study that a former grad student, Waylon Yu, did a while ago now. Just we used a simple model of inflammatory pain where we injected mice with CFA into a paw and then just looked at alcohol consumption. We saw that in male mice, CFA was able to increase alcohol consumption, whereas in female mice, we saw no such effect. And so there's this interesting sort of sex-dependent effect. But the idea is that increasing pain had some ability to drive alcohol consumption. So that's one direction, but it's not just that direction. It's also, here's another study that Wayland did that we've never published, but many groups have done experiments like this. If you take an animal, a mouse or a rat, and give them long-term access to alcohol, in particular, we use an intermittent access approach here. And we looked at, this is Hargreaves, so this is thermal sensitivity, thermal nociceptive response. We see that there is a increased latency or reduced latency to withdrawal. So they're more sensitive to this thermal stimuli. And the idea is, so alcohol has this two-way road, and this is really rings true when you look deeply at the clinical data. And so Katie Wickowitz and many, many others have highlighted that pain is really important, a really important comorbid condition for shaping how individuals respond to alcohol. So alcohol, history of alcohol dependence can increase risk of chronic pain, but also chronic pain can be a risk factor for developing alcohol dependence. So to me, this is an important area of study, and this is where animal models are useful, where we can start to probe the chicken egg question, so to say. What drives what?
That
We can begin
male-female difference,
Yeah.
that's amazing.
It's really amazing. And the sort of exciting thing is I'm reviewing a paper right now that I will reveal nothing else other than they beautifully replicated this data set. And so it was nice that someone else found the same thing. And I mean, I have some thoughts as to why that are entirely hand waving. So I don't know yet, but it is really remarkable. And I'd say we see a lot of very interesting sex differences as we dive deeper too, which I think is important. And so when we think about, the brain, here's sort of a classic view from a review rather from George Koob. I'm just hiding some things so I can see stuff better. From George Koob and Nora Volkow. It's pretty old now. They've probably written 4 reviews like this since then with the same or a very similar diagram. But the idea being that there are different, they conceptualize there are these different aspects to the development of a substance use disorder or alcohol use disorder. We have this binge phase, withdrawal phase, and this preoccupation anticipation phase. And really where I have focused, so we think they have, they conceptualize different circuits contribute to these different responses, and they all sort of interact. And really where I have hunkered down for, sadly, it's about 20 years now, so it's been a long time, has really been deep here in the extended amygdala. And so this is where I have spent a lot of effort and time trying to understand how does this contribute to behavioral dysfunction or, various aspects of alcohol use disorder. And so the extended amygdala, it has, broadly speaking, I'll focus on the 2 major parts. There's the central nucleus of the amygdala, or the CEA, as well as the bed nucleus of the striatum terminalis, or BNST. We dabble in both, but I'll focus on the BNST. So this is an interesting structure. It receives a GABAergic input from the central amygdala. It receives a glutamatergic input from the BLA. It receives other glutamatergic inputs from frontal regions, insular cortex, ventral hippocampus, as well as some thalamic regions. So it receives a lot of different inputs regarding the external and internal state of the animal. But where, the really, I think, nifty part to me is it also projects to all these midbrain, hypothalamic, and hindbrain regions. And that's not a one-way projection. This is a bidirectional interaction. And I think it's through these bidirectional interaction that we can sort of get entrainment or engagement of, the BNST can act as a central regulator for brain-wide states. Like, for example, many years ago, grad student Chris Mazzone did some work where he activated a DREAD in the BNST, and we looked at whole brain metabolism using a PET approach with Michael Michaelides, and really the most robust effects we saw were in frontal cortical regions. So it serves as a way to sort of rapidly manifest changes in behavior. And if we think about what we know, There's sort of classic studies suggesting roles in stress-induced reinstatement, the opioid withdrawal, among other things. There we go. Mike Walker for years had this really elegant series of studies looking at fear and anxiety. And in particular, a group in Japan, the Manami Lab, has done some really nice studies looking at neuropathic pain, and not the pain response per se, but the anxiety or dysphoria associated with it. And so this has been a really interesting thing for us over the years in terms of informing how we think. And then, there's some more, I would call them not modern, but circuit or molecular manipulations that can regulate anxiety from our group and many others. If we think about alcohol, it can regulate the rewarding properties of alcohol consumption. And then a really recent study from Tiffany Will's lab at LSU showed that the BNST plays a really critical role in alcohol exposure-induced pain. And so it's really sitting there at the interface, regulating all these behaviors that are really interesting to me with respect to how pathology associated with substance use, in particular alcohol use disorder. And so BNST is interesting. It's mostly GABA neurons. There's a peppering of glutamate neurons as well, but mostly GABA. But these aren't all the same GABA neurons. There's lots of different peptide markers or peptides in these neurons that, we often use as markers, but I think it serves as they probably play important functional roles as well. And so there's a corticotropin releasing factor. I'll just refer to it as CRF. That's what we're going to dive into today. But also there's a bunch of other really interesting peptides, dynorphin, somatostatin, neurotensin, nociceptin, among others. And so this is just an example image showing indeed there are CRF neurons in the BNST. This is just a CRF AI9 mouse showing mice that express CRF also are expressing TD tomato. So CRF is there, presumably released in some fashion. And then also we know this is a study from led by Catherine Marcinkiewicz, former postdoc, and Chris Mazzone, a former student, just showing that if we put a channelrhodopsin or an opsin in CRF neurons in the BNST, and then look at local connectivity. In here, we were specifically looking at outputs to the midbrain, the VTA. We saw that there's a local GABAergic projection. So these are GABAergic neurons that can release GABA locally and also distally in downstream regions. And we know, just to highlight why, why do we care about this? We know that if we chemogenetically inhibit or if we activate GI signaling in these BNST neurons, we can reduce alcohol consumption. And so here is a paper where we did this. This was led by Kristen Pyle roughly a decade ago, and just showing if we give the chemogenetic, the DREAD agonist CNO, we can reduce alcohol consumption in mice that have the GI DREAD expressed. And also, interestingly, we saw that there was a significant increase in center time in the open field. So that would be, we would say that's, some sort of reduction in anxiety-like behavior or avoidance behavior. And that sort of position these neurons as potentially this area where we can modulate both anxiety-like behavior and this alcohol consumption type behavior. And so, taking a step or looking at some of the other work that's been done, this is a study from the Manami Lab, came out in 2019. And so they did a really nice study where they used a neuropathic pain model recorded from the BNST 4 weeks after cessation of, 4 weeks after they conducted the procedure to induce pain. And they found there was an increase in spontaneous inhibitory postsynaptic currents in the dorsal BNST. So there was increased GABA release onto these BNST neurons. And then what they went on to show was that this increase in GABA release was blocked by a CRF1 antagonist. And moreover, in an awake behaving animal, if they put the same CRF antagonist into the BNST, they were able to get a increase in dopamine levels within the nucleus accumbens. And this was also associated with a conditioned CPP to that CRF1 antagonist. So it suggested that we have these, there's an inhibitory tone on the BNST that is somehow driving inhibition of VTA neurons. So this was interesting to us for a number of reasons. And then also they found a more recent study that came out in Scientific Advances that they're looking at outputs to the lateral hypothalamus, or LH. They found that with this same model of chronic pain, there's an upregulation of activity in CART neurons. So CART is a neuropeptide, cocaine, amphetamine related transcript. the activity of these CART neurons was increased, leading to a suppression of activity in these BNST outputs to the LH. So it's sort of this idea that in this model, we get engagement of some sort of local GABAergic circuit within the BNST that drives changes beyond the BNST. And so we wanted to know sort of an offhand question. We were curious, what about inflammatory pain? So I think inflammatory pain is not as in animal models is not as robust of a driver of, I would say, dysphoria or anxiety as some of the neuropathic pain models, but there have been some spotty reports. And so we were curious, what happens in a model where we can drive an ongoing inflammatory pain state. So we injected mice with CFA, and we saw this was work, Waylon, you did years back, and we saw that indeed we can get a heightened or reduction in the paw withdrawal latency in CFA exposed mice compared to saline controls, and this was both in male and female mice. And so then,
Tom, I may have missed.
yeah.
What is CFA?
Oh, I'm sorry. CFA is conditioned Freud's adjuvant. It is a bacterium that induces a heat-treated bacterium that induces an inflammatory response. Basically, when you inject it, you get like a inflammation and swelling in the paw that leads to that sort of inflammation and swelling goes up, and then it fades a bit, but you're left with this hyperalgesic state where mice are more sensitive in the paw. Sorry about that. Thank you, Dory. So we just want to know what does this do to BNST function? And what we saw was pretty sort of interesting, pretty similar to what the Manami lab saw with the models of neuropathic pain. In the dorsal BNST, we saw this upregulation of GABAergic function or spontaneous iPSC frequency, whereas we didn't see much in the ventral BNST. So it sort of mapped on to what they were seeing there. And this was interesting because this is actually something that we had seen before. And so when one of the studies we did a while back, trying to understand how serotonin actually can drive negative behavioral states. I feel like I presented this when I was like a third-year faculty at UCSF, and Howard Fields gave me many, many, many good suggestions on things to do. And we did all of them, and it was great. But briefly, what we found is that serotonin, when you were looking at BNST function, looking at these outputs to either the VTA or the LH, when we put on serotonin, we see an inhibition of those outputs. So we sort of get engagement of this GABAergic response onto outputs. And also when in vivo we gave an SSRI that would drive increased fear or anxiety, we saw a similar increase in GABAergic transmission onto these outputs. So essentially, we're silencing these outputs. We also saw the same in a model of alcohol dependence, where we recorded, this is now recording from CRF neurons that projected out to the VTA. We see an increase in GABAergic transmission onto these neurons, onto CRF neurons. And then this is interesting to see sort of this conserved engagement of this inhibitory, maybe microcircuit. This is all in rodents. And so we have a long-standing collaboration with Kathy Grant at OHSU where we record from from brain slices obtained from primates, where we found that a history of alcohol drinking led to a similar increase in spontaneous iPSC frequency in both male and female macaques compared to control mice. So we sort of see this generalized difference or this engagement of this microcircuit. And what it is, so this data and others from Eric Dumont when he was in John Williams' lab, Zoe McElliott, her lab, there's a model when you take all this data together that we have GQ signaling in the BNST appears to engage a local GABAergic circuit that entrains, it engages GABA neurons to suppress these outputs. And so work from Carl Diceroth and others found that these GABA outputs can drive anxiolysis or stress buffering or reward. And when we engage these local GABA neurons, we can then increase their activity. And there are some studies showing that CRF-R1 can also drive this increased local GABA release. And what this does, it takes these neurons that are buffering stress or driving reward or reducing anxiety and shutting them off. So you get this engagement of this aversive state. And so this to us was pretty interesting. And we started to think more about it. So we were really focused on CRF. And one of the things that has emerged about CRF, in particular CRF1 antagonists, is that they have failed very robustly in the clinic for a number of different conditions. So I'm just one of the, a moderately recent paper, it's nearly a decade old, but from Marcus Heilig's lab, showing that a CRF1 antagonist didn't appear to have robust anti-craving effects in a population of anxious, alcohol-dependent women. There are some neuroendocrine effects. And there, I think there's a lot to unpack about why this might be and how this might relate. But I think at the end of the day, there was this really nice review about these translational failures. And, my perception is, given all these failures, it might be hard to get a company to want to invest in this area. But also, I don't think that means this is not a fruitful line of discovery. The idea that, perhaps CRFR1 is just never going to be a target in people, but that doesn't mean that this body of literature we've developed in rodents, we need to look beyond that. Are there downstream effects that might yield interesting targets for treatment or biomarkers? So this is sort of an inroad to try to understand that. And the question we had was that, and it's sort of related to this, we do dread work, or when all of us do dread work, when we're manipulating neurons that contain peptides and transmitter, and even, if we're thinking about dopamine neurons, because so many people love those, we're manipulating dopamine, but maybe they also co-release GABA or glutamate. Really, what's doing the business there? Is it, for us, when we're manipulating CRF neurons in the BNST, we wanted to know, is it the CRF or is it the GABA that's having effects here? And so what we did, there are a number of ways to ask this question. So we had a method to block GABA release. It's a genetic approach where we can suppress VGAT expression in a cell type specific fashion. So Bob Messing's done some really nice work using this approach in the central amygdala. So this is just a viral approach. We inject a VGAT shRNA. We also have a CRISPR vector that works. And we also have a method to manipulate CRF release. We have a flock CRF mouse where we can infuse AAV-Cre to suppress CRH expression. And then we also have a CRH CRISPR vector where we can suppress CRH in a CRH Cre mouse line. And there are sort of advantages, disadvantages to both. But we found that they seem to work relatively similar, although maybe not precisely the same. And so this is just to show you VGAT suppression works. And so we put chanorodopsin and either a control vector or the VGAT shRNA vector into BNST CRF Cre mouse and then recorded locally and found, indeed, we see very little light-evoked release when we suppress VGAT shRNA compared to control mice. So this approach works. A number of labs has used it. Zoe McElliott's lab has used it in the central amygdala, found similar robust effects. So that worked great. And the viral approach, we have a floxed CRF mouse that Larry's wife all made. We can put in a AAV Cre, so we used CAMK2 Cre here into the BNST, and we can suppress CRF mRNA in this structure. And I'd say with our viral manipulations, we're not getting total, we're measuring mRNA versus protein often. We're not getting complete deletion. It's not as if there's no signal. We get a knockdown. And I think it's important to recognize, but also I think sometimes this is as good as you can get even with a viral approach here.
Tom,
Okay.
why there is so much variation though?
Yeah,
I
I mean,
understand the 50% knockdown, but the variation, I've never seen it before. I mean, do you see infection or this is the
yeah. So I mean, I think it has to do with variance in infection.
But then you should.
Yeah, so for this particular set of experiments, we did lots of correlations to look at relative expression and see if that related to anything, and it didn't. And so, I mean, I think there is a fair amount of variance that exists. I don't know, we've done a fair amount of knockdown Cre studies, and this is maybe a little bit higher, but not far off. And maybe it's just the function of where we're injecting. It's kind of a weird spot, and maybe we get some virus leaking into ventricle, so it's the local titer is lower. Good question, though. Okay. So that sort of is the heart of the matter. So we want to know what from these cells is driving or altering behavior here, and sort of just marching through these various states. And the big picture idea is, when we think of the BNST, we think it's, the dogma is that the BNST is important for negative reinforcement. And so we were trying to see, testing the broad hypothesis that if we're able to manipulate, if a manipulation is able to reduce pain and reduce anxiety, then we should also see a reduction in alcohol drinking, and sort of we can get a common thread through these that might lend insight into the actual sort of larger role that this system plays. And like similar with VGAT, we can sort of get a through line through these different conditions or pathological states. And so this was published a while back. This was from Wei-Lin Yu. And what he found was that when he used the AAV-Cre and the flox-CRH mice, he did a Hargreaves, looked at thermal nociception, there was a significant increase in the paw withdrawal latency. It's not a gigantic effect size, I think is important to note. And this is a pretty well-powered study, we'll say. And so there's a significant effect. It's a significant increase in the latency. So it's suggestive of a reduction in thermal sensitivity. So it was notable and interesting. And in this same paper, we did some in vivo imaging of these CRF neurons and found some interesting patterns of activity. But this was here, and sort of it was like a lingering question. One of the things in this study we never really did was look at what happens when they're in a more persistent pain state, such as you might get with inflammatory pain. And so actually, before we did that, one of the things Wei-Lin was interested in is this idea that using a inescapable hot plate assay. And so this was Greg Scher had some nice studies. Greg Corder in his lab did some nice studies using this assay to parse out nociceptive versus sensory versus affective behavioral states associated with pain. And so we looked at this, and really we didn't see a lot of effects when we manipulated BNST here. We saw this there's only one significant difference we saw, is that there was this increase in paw tending in female mice when we deleted CRH. So there's this slight change in affective motivational states, but really it was a lot of null findings following lots and lots of really dense scoring that we did. So, part of our idea was that given the Manami work, we might expect a really robust effect on not really the nociceptive effects, but maybe more of an affective behavioral effect. So we didn't really see much though. So then what I was saying, we actually wanted to look and see what happens in a more, persistent pain state. So again, we moved to the CRISPR-based approach, and part of this was driven by, we have the CRH-Cre mice, and we wanted to do an apples-to-apples comparison. So if we're using the CRH-Cre to suppress GABA release, we want to use the CRH-Cre to suppress CRH as well. And so what we do, we injected the CRH CRISPR into the BNST again, and then did the same experiment where we looked at, or sort of a subtle version of the experiment, where we looked at response in mice that received saline injection into a paw, and then mice that received a CFA injection into a paw that would induce a heightened nociceptive sensitivity. And what we found was that indeed, CFA has this consistent effect, although admittedly in the females, there's a lot of variance that we see in the CFA group. And so we walked into this, the hypothesis being that we're going to replicate our findings of the flox CRF. And when we look at the male mice, indeed, there's this main effect of the CRH knockdown with the CRISPR vector. Not really there's no interaction or synergistic effect with the CFA group. But sort of curiously, we didn't see this effect in the female mice. And we've gone on and we've looked more closely at the efficacy of the knockdown or the CRISPR approach in the females. And it looks like it doesn't work quite as well. And we're a little confused why. And so this is sort of an experiment. We're increasing the sample size and trying to understand a little bit better what's happening. And it might be, there are some subtle differences in how the CRISPR vector is working in our hands. So this was, I'd say, it didn't actually yield a tremendous amount of insight. If anything, it didn't show us a really robust differential effect in the presence of CFA. And again,
did you look at a possible correlation with the estrus cycle?
We did it. Yes, that's a good question, Nuri. We did a little bit. We did sort of in some of these mice, we did not look at estrus. But we did an additional set of mice where we did look at estrus, and there was a hint of a signal there. But I think we did the experiment in a way where we housed in this, like in that particular experiment, we housed the CFA females with saline females. And there's a bit of a confound there where there can be social transfer of pain behavior. And so there's something interesting, but it's really hard to interpret. So there's a little bit of a hint there, but it's one of the things we're in the process of revisiting that data set, trying to understand, because that when I looked at this as well, it made me think estrus might be playing a modulatory role there. And I know former postdoc from my lab has done some recent work showing that there's a really, really interesting estrus BNST CRF interaction that happens that regulates alcohol drinking. So it sort of makes it wouldn't be a leap to think it's also happening in what we're looking at. So this is definitely a follow-up experiment. I would say we also looked at that inescapable hot plate assay. The slide's hidden, I realized, because I clicked next. But again, we saw absolutely nothing in that data set when we looked across the animals here. So it sort of was not particularly interesting. So then we wanted to look at GABA release. And so this is where it sort of a non-result became more interesting. So we use this VGAT approach, and I'll just, in the females first, what we saw, let me get this going. We really saw no effect, not even remotely, when we suppressed VGAT release, or we suppressed GABA release in females for BNST CRF neurons on the nociceptive response. So that was an interesting non-result, especially given when we looked in the male mice. Again, we found in both groups, we found an effect of CFA, but in the male mice, we saw that there was this robust effect on, or a robust reduction or mitigate this CFA effect on thermal sensitivity. And so sort of shown here that in male mice who received CFA with the scramble vector, we have this sort of robust reduced latency of response, whereas those that got the VGAT vector did not have that. So it suggested that there's something about GABA release, regulation of GABA release from these neurons that's differentially regulated in male and female mice. And this is interesting because the data I showed you earlier showing that there was a CFA effect in the BNST was actually all from male mice. And so we have a smaller data set with female mice where there doesn't really appear to be an effect. So it suggests that perhaps there is some sort of sex difference in this inflammation-induced plasticity of the system that may be behaviorally relevant. And so again, here we use this hot plate test to look at changes. And really, there's nothing that happened here. There were no differences that we saw across any of the behaviors that we measured. So it was sort of a null finding here. And so putting the pain together, we had this, I'd say, earlier data set that suggested CRF deletion can reduce this thermal response in both sexes. It may be using the CRISPR approach, we really only saw it in males, so it might be a more robust effect in males. But also, when we suppress VGAT in these neurons, we only really saw an effect in males and nothing in females. And it was reminiscent of some data we had found and published where
We lost.
We lost Tom. Are you back?
Oh no.
I think you're muted right now, Tom.
Hear you.
there we go.
Great.
Sorry.
Good to have you back.
I'm back. But we saw, so briefly, we saw a sex difference where when we manipulate
We
dopamine
can't
signaling.
wait. We can't see your slides now.
Oh my gosh. Thank you, Dorit. So many buttons. Thank you. You got me?
Yes, it's good.
Okay. So the sex difference we saw was reminiscent of a sex difference in dopamine function that we published 4 or 5 years ago, 4 years ago, maybe. So it was kind of interesting, suggesting maybe BNST in males might play a more critical role in regulation of pain-related behaviors versus females. And I'm not sure why, but it's sort of it seems to be somewhat vaguely consistent through what we've been doing. And so then we were curious about, I wrote anxiety and fear, but the fear is very lengthy and complex, so I took it out, but just trying to understand the role of these systems in regulation of anxiety. And this is where it's pretty easy to talk about. So we deleted CRF in the same way. We saw no effect in the open field, no effect in the elevated plus maze. We looked at novelty-induced suppression of feeding and saw no effect with deletion of CRF. And then we looked at startle response as well and saw no effect of CRF either. And so this was sort of to us, a very surprising non-result that when we deleted CRF, really nothing happened to anxiety or aversive-like behaviors. Importantly, at the baseline state. So I think that's probably an important caveat to think about there. And so then we looked at VGAT suppression again, and we didn't do this across the whole panoply of experiments again for various logistical reasons. But when we looked at open field, we saw no effect in male mice at all. But when we looked at female mice, we saw this increase in center time with respect to the scramble control. And when we looked at the elevated plus maze, we saw a similar effect here, where there was an increase in females when we suppress GABA release. There's an increase in open arm time, no change in closed arm time. So not a locomotion effect, but really an avoidance-like effect. So it sort of started to, when we looked at manipulating these cells, in the females, we saw more of an anxiety-like effect, where in the males, we appeared to see more of a pain-like effect. So this was somewhat interesting, and it made it a little bit more confusing, sort of thinking about
Lost you
why.
again.
Oh my gosh. Am I gone?
No, I can hear you fine.
Oh, okay. Thank you. I'm at work too, hoping UNC's Wi-Fi is working. It might be state cuts, you never know. And so we saw this differential effect where more robust effects on anxiety in female mice compared to male mice when we manipulated CRF VGAT release in the BNST. And so then what we were, sort of how does this relate to alcohol consumption? And so what we did here is, we do a lot of work where it's home cage drinking, but in this particular approach, we wanted to use something that would yield a little bit more insight, give us a couple extra metrics. So we used operant self-administration, and we used an approach that led to really high intake. And so we used mice, so our controls received 2% sucrose for each reward. And then we had mice that received 2% sucrose plus 9% alcohol. That is the equivalent of a dry Moscato. If anyone's had dry Moscato, it's kind of gross, but the mice seem to really like it. And so there was they had a mild food restriction. They were group housed. The experiments were done in the dark cycle, and we did everything in male and female mice.
Just to clarify, Tom, you're not actually giving them dry Moscato, right? You're just giving them...
No.
Okay, just checking.
We should maybe. Bet it would be probably taste better than sucrose and alcohol. And so we look at this is a 1 hour exposure or 1 hour session. We can get extremely high levels of alcohol intake. So we sort of, we decided we wanted to get really high intake because the idea is that really high intake engages some of these systems, and repeated really high intake. And when we looked at, these are the grams per kg that they drink. And then we looked at the blood alcohol level rather these are in the C57 mice as we were getting this particular model up and running. You can see that there are extremely high levels of the blood alcohol content in these animals. So they're all well over this sort of demarcation of 80 mg per deciliter, which is equivalent of 0.08 in people. So this is a very high level model following 45 minutes of self-admin. Okay. And so then the way we did these experiments, we did 9 days of FR4 self-admin. We did 2 progressive ratios in part because the literature had multiple methods that people had used to look at progressive ratio, both arithmetic and exponential. And we wanted to be able to compare to established work. Really, there's not many differences between what we found, so I'll just focus, I think, on exponential. And so first to show you, so we focused.
I want to understand. So
Yeah.
they press the lever for both sucrose and alcohol,
No,
right?
okay, I'm sorry. So each group only gets one type of reward. And so we have mice that only get alcohol and sucrose, and then we have mice that only get sucrose. Yeah, it's not a choice procedure, which is, thank you, Dory, important distinction. And so we centered our injections around the ventral BNST, but really this is a BNST knockdown. And so in female mice, what we found in the Cree mice, so CRF reduction, there's a significant reduction in responses per session of the active lever. And so they poke or they lever press less than you will see in Cree mice compared to GFP mice. And there's not a significant effect, but a sort of a close effect that we see when we look at the grams per kg that they consume here, sort of the calculated amount that they would consume. So there's a hint of effect in females when we knock down CRF, which was interesting. Then we looked in male mice, same exact experiment. These are alcohol sucrose animals. We saw that there was this incredibly robust reduction of both responses for the active lever as well as the grams per kg that they consumed. And so really this was I think we were honestly pretty surprised at this result. I anticipated no effect here, and my hope was to stop looking at CRF, but this really got us back into it. So this was pretty exciting, but I think one of the important confounds is we're looking at alcohol and sucrose together. And so we need to also think about what sucrose does. But before that, we also just looking at progressive ratio. So this is, the mice have to press, they sort of press more and more times until they reach a break point, and that's when they stop pressing. And we see that when we delete CRF, the mice their progressive ratio is lower, so they will press less to get alcohol. So their motivation to press to try to get alcohol is reduced when we get rid of CRF in both males and females. So this was sort of consistent what we saw, and interesting that CRF from this relatively small population of cells had this really robust effect. But again, we had sucrose, it was alcohol sucrose, so we need to look at sucrose as well. Importantly, we saw no effects with sucrose, with the knockdown of CRF on sucrose responses per session. So there was no difference between Cree and GFP when we looked for sucrose alone. And I would say this is in a separate set of animals. These animals never saw alcohol. We wanted this to be a really sort of, for lack of a better term, a really discrete experiment where we looked at this. And we also looked at progressive ratio, and there was no effect there either. And so that was honestly it was the opposite of what I thought. I thought CRF would do nothing, but the data is what it is. So that was interesting. So we wanted to look at what about VGAT suppression? What will that do on this behavior? And again, the same exact type of behaviors, same exact behaviors actually. And so in females, again, we really saw no effect. This is looking at responses as well as grams per kg. There's like a little hint of a reduction here, but there's no significant effect. This was pretty surprising to me given the effects on anxiety-like behavior. When we looked in males, what we saw was a little different. There was a main effect of the virus, so knocking down GABA release from these CRF neurons led to an increase in alcohol consumption early on, and then they sort of hit this ceiling effect over time. And we see a similar trend in the alcohol consumed, and we didn't see any effect for progressive ratio here. It was sort of this tantalizing, sort of vaguely up in both males and females, but not significant. And so then, of course, we wanted to look at sucrose, just for a full appropriate design. And again, we didn't really see much for sucrose when we're looking at the FR4 responding, so 4 lever presses to get a response. Really nothing in males or females. However, when we looked at progressive ratio here, we saw that there was this main effect of VGAT suppression across both males and females. So the idea that, it didn't affect this, FR4 is, I would call moderately low effort. You have to hit a lever 4 times. But when you make them work a little bit harder, so they have to press more and more over time, the VGAT suppression led to a reduction in effort expended to do that. And so this was pretty interesting. And CRF had this really massive effect in both drinking and motivation in both male and females. And this VGAT suppression in males increased drinking early, but in both males and females suppressed sucrose motivation. And I think really the way we did experiment probably obscured some of the effects on drinking, because if sucrose, the idea perhaps there's this subtle reduction in motivation to consume sucrose. If we have an alcohol sucrose solution, that sucrose may be masking some of the effects that we would see on alcohol. So a nice reviewer pointed that out, and it's a really good point. So it'd be interesting to follow up. And so then when we sort of take this all together, the idea is that the BNST is this really critical structure for anxiety, dysphoria, pain, negative affect, negative reinforcement. And what we found when we think about sort of the idea of anxiety as potentially a driver for some of these effects, it didn't really seem to hold water with what we were looking at. Sort of the big surprise was CRF didn't really do anything with anxiety. It was, there is this potential where, when we think about pain and alcohol, that there is some sort of synergistic effect, and perhaps these CRF neurons in the BNST are potentially a means by which CRF is able to drive heightened alcohol consumption. I think that is definitely sort of an interesting idea. And, I think the logical follow-up is to do some experiments looking at how pain-enhanced alcohol consumption might be regulated by this system. But I think also when we think, there's this sort of model where there's increased GABA release on this output population of cells. I think what this data tells us when we look at the VGAT data is that really when we look at the affective sort of performance or the anxiety-like behavior, it's probably multiple. It's probably not just CRF cells. Maybe it's a other population of cells within the BNST, and maybe even it's another output, such as something from the central amygdala. And there's data to support that model. And really, there's some intriguing sex differences that we don't understand. Whereas males, there appears to be more of a pain-like phenotype, whereas in females, there's more of a anxiety-like phenotype when we think about pain versus anxiety. And that might actually be something that relates to sort of inherent motivational states that we see for drinking in males and females. And I think one of the other things is that, there's this real dogma that CRF in the BNST is anxiety. And our data just didn't really support that. And, we've sort of come around to this idea it might be more important for arousal, salience, and novelty, and not just aversion. And we started digging into that a little bit. So here's the model sort of thinking about different aspects of GABA or CRF signaling. But we started to think about novelty, and we started, trying to understand what are the best ways to look at novelty. And we've done lots and lots of different experiments trying to do this. One that has given us some nice control over delivery of a novel sort of thing is, here's an experiment we have, an olfactometer, where a mouse can stick its nose in a port, and when the scent goes on, they stick their nose in a port. So we have a nice time-locked response, and we're recording from BNST CRF neurons here. And so we can see with this specific odor that the very first time they get this odor, it's a novel odor to them, we have this nice big response, but then over time it sort of degrades. And there's this degradation where it's no longer novel, and we're sort of intuiting that this might become less salient. It's sort of, I know what that is, I know what that is. And so the odor here, these are male mice. This is actually female mouse urine. And so male mice really like that. And the idea, there's sort of a desensitization, for lack of a better term, of this response over time. This is some data from Lily Koyman, a talented technician lab that has been working with Allison Rowland, another faculty in the lab. So we're sort of excited trying to understand the role of the BNST in perhaps not just anxiety, but more arousal type states. And then another thing we're really interested in is, CRF is having a really robust effect. And then I don't know if any company will ever want to look at a CRF1 antagonist, as I said before, but how does this propagating throughout the brain? And so the BNST has lots of different outputs to the VTA, the lateral hypothalamus, the PAG, the dorsal raphe. We've done some work with Todd Thiel sort of highlighting the VTA as a potential locus of this regulation of drinking. But we also have a pretty deep dive, we're really interested in serotonin signaling as well. And we have some really new data suggesting that when we delete CRFR1, so now focusing on the receptor versus the peptide itself. So when we delete CRFR1 in the dorsal raphe, we get this, I'd say this is a nice hint of an effect on alcohol consumption in female mice. Not amazing. But what we also have started doing in the lab is trying to look at not just alcohol consumption, but also what happens when we have sort of a conflicted alcohol consumption. So you put a little quinine in their solution. Normally mice don't like, quinine's bitter, they don't like it, they will not drink as much. And we did that in this, again, this is like pretty new data, who knows what'll happen, but it's still exciting to us. When we look at that quinine suppression of drinking in these same mice, we can see that when we get rid of this CRFR1, we have this really robust reduction of consumption. And we have I'd say this is sort of part of a larger serotonin project we've been developing, looking at cortical serotonin signaling and I'd say models of aversion-resistant drinking. So it sort of makes sense with what we're thinking here. So it's a pretty exciting idea and perhaps highlights that, maybe thinking about serotonergic targets might be a useful downstream signal that could come from all of this work on CRF and CRFR1 signaling. And with that, I'll say thank you so much for your time. I apologize I went a little long. Again, all the folks in the lab who contribute to this as well as our funders. Thanks so much.
Thanks, Tom. That was great.
Yeah.
We have time for a few questions, I think. So I don't know if I should just be unmuting people, or they should just unmute themselves.
Let me ask a quick philosophical question, Tom, about why do you think the animals drink more when there's a painful signal coming in? I mean, from an evolutionary standpoint.
Yeah, I mean, I think alcohol has analgesic properties.
For sure.
So my guess, I guess my first suspicion would be that if you're in a heightened pain state, then something that's analgesic, you'll consume more to sort of relieve that pain.
Sure. Okay.
And we have data sort of supporting that model where we can look at not sort of the inflammatory pain, but we can see that alcohol consumption can normalize nociceptive responses after extended access. So I think I don't know, I think it's a good question because there also could be a bunch of sort of analgesia independent mechanisms at play too.
So Tom,
Yeah.
kind of following up on that, you have so much experience working on so many different behavioral models that try to probe dysphoric states. I mean, what has it taught you about dysphoric states more broadly? Is that really a useful concept? Is it something that we should be not, continuing to hold together, lump together, or should we be fractionating it? What's your sense right now?
I think that all dysphorias are not equal is probably how I would conceptualize it. Is that, it's nice to sort of lump everything into there's a generic bad state, but I think that different things will produce different bad states. I think opioid, in particular, like opioid withdrawal, acute and even protracted, will produce a very different behavioral state, and I think brain state compared to, say, alcohol. And so, a lot of it probably has to do with sort of PK, pharmacokinetics and how different things happen. I think there are some conserved aspects, but I also think it's maybe a good opportunity to use, it's a good opportunity to be more precise about what we look at. And I think part of the, one of the great issues or, one of the areas of growth for animal research is that it's not very frequent that you see a person who will only ever drink alcohol. And so I think, this is an important area of growth that, let's pretend the NIH is well funded and there's not things happening. To me, the idea of looking at more, sort of human pattern driven behavioral models would be extremely important, but also it's a real rough road to go down because if you submit a grant where you're looking at two things, you get someone who likes one thing, but doesn't like the other thing. You get another person who's the opposite. And so you just kind of you catch it from all angles. And so I think it really is something that's important that requires programmatic high-level intervention. I don't know, I think it would be a way to parse out some of these states more efficiently.
Can I ask a question?
No.
Actually, two things. Number one, regarding what Howard says, is it possible that pain increases anxiety and then anxiety increases drinking? Number one. Number two, you don't need the sucrose, well, for operant self-administration, and it really may be masking. Mice will self-administer alcohol alone, and to get to test anxiety after alcohol use, it would be very interesting, and we know that is true, to get the mice dependent on alcohol using vapor, and then you see the negative effect of alcohol, and there you do the manipulation. And I bet you anything that you will see interesting phenotype.
No, I agree with you. So point one, yes. I mean, I think pain does not exist in isolation. There is this important interaction with affective state where this is where it can be a little muddy. You can get anxiety-like behavior, but you can also get more generic dysphoric-like behavior with pain. And I think that is part of the lens with which you have to look at some of the behavior. So I agree with you there. That's an important aspect. I would say when we've looked at so for CFA, where we saw an increase in alcohol drinking, we never really have seen robust classical measurements of anxiety-like behavior. I would say I've seen more recent studies that use different approaches that do reveal things, some different assays. So I think there's room for growth, for our growth there. With respect, mice will drink without sucrose, absolutely. I don't disagree with you. I think it's definitely a limitation of what we did. I mean, in a perfect world, we could snap our fingers and do things differently. Maybe we would have. But that's what we did, and that's why we did the control. I do think, we have done a lot of experiments looking at, so alcohol vapor can induce alcohol dependence, and you'll get increased alcohol consumption. And the idea is that is, thought to be probably the best model of negative reinforcement. And so I think that would be an important area to delve into here for some of the things that we're studying. And so I don't disagree with you. We're doing some work there with some different systems, different parts, like entirely different amygdalar regions, like not central, not basal, not BNST. So we're working on that, and I think it is a very robust approach, which does model negative reinforcement the best. I'd say we've done some experiments where we've looked at CIE and what happens when we manipulate CRF neurons. And we've seen mixed bags of response with respect to some of the anxiety-like behavior. We didn't do the CRF manipulation in that model, and I do think that would be interesting because could be that, when we looked at anxiety, we sort of looked at baseline anxiety, not sort of a pathological state. So it might be revealed there. I think that's a valid and important point. We have done a lot of work sort of looking at different models of fear, predictable, unpredictable, ambiguous, and manipulated CRF when we've done that. And we've seen no effects on anxiety-like behavior post-fear that were by manipulating CRF. We actually saw opposing effects, where when we deleted CRF in female mice, we saw actually increased hyperarousal. And I think part of the devil in the details is there is CRFR2 signaling in the BNST, and CRFR2 is important for sort of stress de-escalation. So if we're killing CRF, we're knocking down CRF, there is the CRFR1 signal that we think, it might drive certain states. But also, if the CRF2 is sort of a different signal that is de-escalating, it might, I mean, it might be a signal to noise issue even, where CRF is doing two things on two different timescales, and when you get rid of it, you don't see any difference. So it's all points well taken, and I think good areas for discovery and advancement.
Well, so many interesting directions. I think we're out of time, so let's thank Tom again for coming and presenting to us. So thanks again, Tom.
Thanks for having me. Nice to see everybody.